discussion, we have allowed people to unmute themselves and speak out. So yeah, I guess we can start off with a recap of what happened in the first BOF quickly, and then yeah. Did you want to take it from here, Dan, for the?
Sure.
Yeah.
So on Monday, there was an initial session that Atul had put together, and we discussed visual impairments and how those sort of end up relating to looking at scientific data. Our discussion covered a bunch of potential action items, but I think Atul and I were both super pleasantly surprised by the number of people who came, but it also meant that the original idea of how the meeting would go was not ultimately how we were able to do it. So we figured another session today where we could go through some of the action items with a bit more interactiveness might help us actually like get a path forward to make some of these things happen. So on this slide are some of the ideas that either came up last time or that Atul and I had sort of discussed afterwards on our own. But they're all ones that we figured we could make some actual progress on, and I'll just stop sharing these slides that we put together for a quick second to jump to the Google Doc, which I've shared in the chat for this session. There's a link to it there. It has sort of an overview of things that were talked about last time and questions that were asked with some of the answers, some extra useful links to resources that we've compiled. But then at the end is this sort of table where you can sign up to help out with some of the ideas that I have on this slide. And you can see Atul and I and a few other people have already done this. But yeah, back to the slides. For each of these ideas, we kind of figured we would go through them one by one, but depending on how things are going and sort of the timing, maybe we would end up wanting to do some breakout sessions and sort of teams of people that want to work on each individual one might be able to group up and make some progress. But the ideas that we had were trying to make palettes that are color blindness or vision impairment accessible to be more of the default, to have a set of resources and guidelines that could be posted somewhere on the Bioconductor website that give people tips for making their packages more vision impairment friendly, building some sort of accessibility score to let people know if their tools are accessible, and then perhaps this could even be built into BioCheck itself. We talked about, I think this was Atul and me afterwards, I can't remember if we discussed in the session, but maybe a version of sort of like GG Repel where colors that are really similar would be made more distant and sort of just repelled away from each other. And I think there are a few different potential ways that could work. But also, we figured we could go through some ideas of new visualization possibilities of how we can augment plots with non-color options. And then another idea was to encourage the generation of alt text, or sort of like text that really describes everything that's important and going on in a plot. And maybe there are ways that this can be done in sort of an automated way. And yeah, I think with that, we can jump into some of the examples. if anyone has any questions, again, I'll just repeat what Atul said in the beginning. We set up this session so that everyone comes in unmuted, but we can all control our mutedness, and you can just unmute yourself if you want to say something or ask a question. So for the goals of making accessible palettes to be the default, or putting together a set of bioconductor resources or guidelines, we were thinking that we could, basically with the bioconductor resources, we could encourage package developers to make palettes just be the accessible palettes be the default for their package. But another idea that I think has been discussed is Ditto-Seek's colors. So the package that I made, and I can go through those features, but I made basically a color panel that is color blindness friendly, but also sort of I extended it to be more than just the 8 colors that it had started with lighter and darker versions, which isn't sort of perfect, but at least allows grabbing of a little bit more, allows the palette to still work with a little bit more complex data. And I don't actually know how to turn this into a more like globally available palette, but I was thinking one of the sort of action items could be doing that. And if, say, one other person who's maybe made a color palette that's really compatible with ggplot could help me to make it, that would be awesome. And then we realized that last time we kind of ended up focusing on color blindness, just probably because that's what Atul and I both suffer from. But there are plenty of other visual impairments. So we wanted to make sure that we open up for other ideas because, for example, someone in the chat from the previous session had mentioned that they're not colorblind, but they have a permanent double vision. So getting input on how we can make plots better for this type of impairment would be great, because I'm sure what needs to be done is different.
Yeah, and yeah, just to, I guess, add further to that, as in even Ditto-Seek's color palette is likely something that is optimal for Dan's and my kind of color blindness, which is definitely the most common one, but there are less common versions where that might not work. So we might still need to update that or modify that to be, I guess, the most general color palette that can be used as a default. And again, the onus is that, yes, having these color palettes as an option helps, but having that as the default really helps, where basically anyone who would use your package would end up with a colorblind friendly or accessible image by default. We're not sure if the person who made this comment about the other visual impairment has joined, and if they wanted to contribute or say something more about that. But we can, yeah, definitely move on. If feel free to add if at any point you think that we haven't covered something, or yeah, if you just had any questions or any more inputs, feel free to unmute yourselves and jump into the discussion.
So for, I think this was your slide, Atul, so.
Yeah, so I guess the next kind of bullet point was having a score, accessibility score for a figure, right? And part of that would also be accessibility score of the color palette that you use. And similarly, the application after that was some sort of a function which, like ggRepel, tries to keep colors which are similar to each other apart in the figure. So the ideas basically are to generate a color similarity matrix for a color palette. So let's say if you have a vector of colors, then you get a matrix of basically how distinct is each color from every other color in the vector. And the common way to, I guess the most trivial way one could think of is just to look at the RGB distances between the color, but that is not perceptually uniform. So there are basically standards called CIE-LAB, which is a representation of every color in the L stands for lightness. A is basically an axis which goes from red to green, and B is an axis which goes from blue to yellow. So in these three axes, it represents every color, and basically that is something which is commonly accepted as perceptually uniform color space. So basically a Euclidean distance in that space is accepted to be consistent with the perceptual distance between colors in any direction. So that could be used. So we have, as in there are resources already available for that. And the question would be, yes, we can make a color similarity matrix for normal vision, but if we wanted to make that for color blindness, specific kinds of color blindness, we could use color blindness filters, which are already available in a couple of packages. I think color blindness is one package. I believe colorblind R is also another one, right, Dan?
Yeah.
And as well as I think color space has functions which tell you what how a deutan, tritan, or protan might see a color that you want to pick. Is that right?
Yeah. So those, one of the color blindness features that I mentioned that I built into DittoSeek was a simulate function. It really the way it works is it just takes the color panel input and uses these functions to convert them into a version of the colors that simulates what a colorblind person would see.
Right.
So I think what we could do would, there's got to be a matrix built in the background, the back end of those functions that we could use for
as in
this purpose.
So the hope is that basically these color blindness filters are, say, accurate enough or good enough. Someone has raised a hand. Please feel free to unmute yourself. And
can you unmute yourself, Leonardo?
Yeah, sorry. I was just curious if there's a particular website you recommend where you can maybe upload like a PNG or a PDF and like see how your current plot looks for colorblind.
Yeah. So I think the Colorblinder package reports that you can do that, and it basically, if it's a PDF, if it's a ggplot figure, it just sort of can take that as input and convert it.
But like so that is great if you're in the process of making it. But let's say you made a plot like, I don't know, a year ago or something. You just have the PDF around. If there's a particular website you would recommend. Like so it doesn't have to be an R package.
There are definitely screen filters which do that. I think, what do I use? I think I use VI Sense or something like that, which basically, and there are a number of filters which we can definitely look them up and update them in the document.
I think there was one shared last time. I'm gonna, how should I share this best? I think in,
anyway, so while Dan looks at that, I'll probably continue with this. So once, let's assume that we have a color blindness or rather a similarity matrix for colors for different color blindness types, then the applications of that could be to just as a whole say how interpretable a color palette that is being used in a figure is. So it could recommend to use a different palette depending on how many colors you're planning to use, or even within a colorblind friendly palette, it could tell you which of the, say, 4 out of 8 colors that you should pick for a particular figure should be. And the second application would be what we call, I guess, the ggRepel equivalent of for colors, which is, say you have a Disney plot or a UMAP plot and you want to assign different colors to different clusters of cells in a figure. And you want to ensure, as in, for example, let's say if there's orange, green, and maybe some weird form of yellow, I guess it might get really tricky for colorblind people to identify where one ends and the other starts. So an easier way would be to, if at all we want to use these colors, we would want to separate them in space and in between have a color which is contrasting with the two similar colors in between. So basically the function would be similar colors are pushed further apart and dissimilar colors are pulled together in the final, say, color assignment in the figure. So yeah, so basically these are the links which I think would be useful, where the first one converts colors from the RGB scale to the lab scale. The second one is it calculates the color difference. So if you go to Wikipedia and type color, search for color difference, it tells you, I think, 4 or 5 different methods of calculating the difference between colors, starting from just the Euclidean distance between the RGB values. And so I think the delta E is one of those, basically, which is the distance in the color lab space, which is well accepted as the perceptual, or closest to the perceptual difference between the colors. And then the last two are basically functions which would help us map from, say, a normal vision color space to a colorblind color space, a deutan, tritan, or protan color space. Did anyone have any questions on this, or any comments if you have worked with colors extensively and as in illustrations, or if you probably know more about lab space than Dan and I, feel free to chime in. Okay, did you find the website, Dan?
Yeah, I posted in the chat.
Okay.
Samina, you should be able to just, yeah, you're unmuted now.
Yeah. So I have, and sorry, I missed the original session, so maybe this was discussed then, but how much would it help to just also have like redundant ways to show different classes, for example? So having like colors, but then also having points. I often find for myself, I'm not color vision impaired, but I often find that that's helpful, especially because sometimes journals will, like if it's too many colors, it's kind of hard to tell them apart anyway, right? And then journals also, sometimes they have like, sometimes they still have like black and white. You have to pay extra charges depending on the journal. And I know that that might not always be possible if it's a heat map, but just curious on that.
That is a good point. And I think the next couple of slides will address that as well. As in, so we have shown a couple of examples how it could be done. So this is basically work that Tejas Guha is a member of, as in he's a summer fellow in the Feick lab, and he's been working on this with me. So so this was basically data just picked from, I think Ruben Trice had shared a bunch of spatial omics data. Or I think this is, immunology data, where it's just, this is showing you different, I guess, tissue types or a cell scatter plot with different tissue types. And the only qualifier or between the tissue types is the color of these 4 blotches of cells, right? And I don't really have any problem with the choice of the colors here. They're quite distinct, and of course there are only 4 cell types or 4 tissue types here, so there wasn't too much, as in it wasn't too hard a problem to find 4 colors. And I think this was probably anyway something that they just chose. So so what we wanted to do basically is just to add a second layer of information, where instead of just having colors, you can just augment the figure with texture or patterns in this case. Or let's say if you have a sparser scatter plot, you could say point shapes where you could say blue triangles instead of, and red squares and yellow dots or something like that. So having a second layer of say information which reduces the dependence on color alone helps, as in I think it helps both in case of as in definitely in case of color blindness, but even let's say if once you get into 20 colors, even for let's say vision normative people, it would help to compare say 2 shades of yellow based on what the overlaying texture or pattern on those is. So to give an example, as in for this particular figure, we didn't even, as in once we introduce the patterns, we don't even need there to be basically color as a distinguishing factor. As in even if, let's say I chose any one of these colors, I would still be able to identify which cell belongs to which tissue type. So even if, let's say this was printed in grayscale, the texture will still be readable in that.
Yeah, and I'll say things like shapes are definitely helpful, but I think it becomes impossible to tell shapes apart when you have points that are overlaid on top of each other. So that's, Atul showed me this representation and I immediately jumped on it. And
yeah, so as in, I guess my initial thought was to try with shapes first, but for a plot like this, it wouldn't really matter because, or it wouldn't make sense because the I guess the orange blob here, right? As in you wouldn't be able to make out whether what shape of points or what point shape it is actually made of. So we definitely wanted to have some texture in that, but we also wanted the texture should be such that it perhaps also works for individual cells. So, and as well as areas of basically denser cells packed together. So,
a question here,
yeah, sure.
Which is one of the things that we started realizing when we started breaking apart some of the Seurat imaging is that they use scale to actually reduce, to make the clusters look better.
Mm-hmm.
So the ones that aren't part of the cluster that are underneath, they're actually made smaller. So it looks like you've got more homogeneous clusters than what you have. So in this case here, you may well have other cells underneath. This may be histology, but if this was a Tisney or a UMAP, you'd have other cells underneath these, and you wouldn't be able to see the heterogeneity within the cluster very easily. So how can, and that's one of the main reasons why people use these approaches and all these colors and everything, is to try to estimate the heterogeneity. Mm-hmm. Now, one way is to like put ellipses around it so you can see, like, you know, whether 90% of the cells are actually being caught in that, and you can see if one particular is straddled. But if you're using this approach, is there another thing that you can add in to indicate the like the dispersion within the cluster?
I think so, yeah. As in, we definitely can, in the sense that I could also imagine basically having, say, a fainter version of the same texture covering the entire, let's say, the convex hull of the cells, or say an ellipse kind of thing, where basically you could still have the ellipse be there along with a texture on that.
Is it worth doing the kind of, I don't know, the New York Times approach, where like you've got like something coming out of the figure reflecting what the proportion of cells are that are colored, that? So you could potentially, I was just thinking, where you could say other there, you could say 45% this, 30% this, 10% this, or actually colored this color.
In DittoSeek, I more recently built some like hex spinning versions of my scatter and dim plots. And with those, you can make a plot where you, for each of the bins, it'll show what the max label is. And then you can make another one where it actually shows, with the same like discrete groupings, the proportion within each bin that were part of that max color or max grouping.
Can you do that for all of the little hexes? Because there's a lot of them.
Yeah, I don't...
so your hex binning would be actually binning, right? It would not be a per cell thing, so it would be
Right, it's binning.
reading and.
I don't have an example prep to show you, unfortunately.
I think you had shared something on BIOS, as in.
Your DittoStick talk was great, by the way.
Thank you. I guess since it is relevant and there are people who weren't here, I'll go through some of the ideas that I have built into DittoSeek just really quick.
The other thing I missed the very start of this. I actually went to grab lunch. I was hungry. I was trying to think of, actually I'll talk later. I'll let you go through your slides first.
Well, it'll be a sec before I bring them up, so. If you want
I think there's only 18 of us in this, isn't there? So.
Yeah, I mean, if everyone wants to just unmute and talk whenever, I think it's small enough for.
Yeah, and you have control over your mute buttons.
Yeah, it'd probably be good if people could just talk rather than it being. I feel nice about the ability to talk. I've been listening for a long time. I find that tough.
Dan, do you think the next two slides might somewhat answer what Aiden is asking about, basically showing to what extent, say, the underlying cells, or as in that would still be.
Oh yeah, yeah. Let me just go through these because I have it prepped and ready now. But so basically, I already talked about the colors themselves. I made it so that legend symbols are enlarged by default, which is it's weird ggplot code, but it's pretty simple to do. And then color alternatives like letter overlays, shapes, or faceting are all great. We talked about shapes, but faceting, you could have, say, 5 colors for 10 different groups if you just have 5 groups in one plot and 5 in the other. We talked about having ellipses go around the groups, but having labels directly on top of them means people don't have to necessarily match the colors from the legend to the group. You just automatically know what that sort of color right there represents. And then that's really everything that I built into DittoSeek. So
one point I would like to make regarding that is, as say the point size changes, as in if you have, let's say, even if you had a large point size in the legend and much smaller point sizes in the actual figure, perceptually it could look different, at least to us. I don't know if it looks different to color normative people as well, but definitely for us, where basically we could mistake something for some other color when it's really tiny. But if we zoom in, or if we basically just peer
if the point size is larger,
very carefully, we realize that it's a different color.
One of the things in very early microarray data is where I'm kind of probably showing my age. I got involved in this project, and what they did was they wanted to make the point at the time that people weren't making their data available publicly and it was hindering research. So what they did was they took one high impact journal, I think it was Nature Genetics at the time, and they took 1 year, right? Then basically the search was basically all the array platform, all the array publications from the year. And there was something like around, I think there was about 70 of them or so. And then it was pared down for ones that actually produced data and ended up being around 40 or 50. And then we divided it up into teams, and people, basically each team got 6 papers and the job was to reproduce figure 1. And then we were able to go through that and actually provide statistics of how many papers had been published in the journal in the previous year that actually provided data, provided methods that were doable, and it was basically a reproducibility paper. And it had high impact at the time because, first of all, and became one that basically people could cite as an instance, this is a problem in the field.
Right.
And sometimes something like that has more impact than, like, there's been a couple of people saying that this is important in the field and saying like how I can't see this.
Yeah.
But unless there's like some sort of data saying to what extent that's a problem, you don't get the journals behind you actually necessarily making those changes. So is that something that could be done for this? Like I don't care what journal it is, but something such that if you did it, it would actually have an impact. And just go through the last 6 months where they had a TISME or UMAP plot, which is just do a text search of the journals, pull it out, and then look at it and say, how many colors do I see?
Right.
Or take it through one of these machine learning ones that will actually quantify the issues with that figure.
Mm-hmm.
And then you actually have some data to say, you know, of the XXX figures published in XXY journals in the past year, basically these are not accessible in 50%, 70%, 30%, whatever it is of cases. And you actually have that data, which then of course you can use like as pilot data for grants. If you want to actually work in this area, it can be used in, you know, when you actually want to give a talk about it, because you actually have like data rather than just anecdotal, I find this difficult, he finds this difficult.
That makes a lot of sense. I've seen a couple of, I think one conference sponsored by IEEE, I think in computer science, they did something, but for accessibility in general of papers. So in terms of how screen reader friendly the papers were, whether their figures had alternative text or not, and all of, so which was basically done for conference submissions where I guess the deadlines are probably much shorter. So people don't care a lot about these things. But if Leonardo, if you have, if you want to jump in, you can unmute yourself, and or is this from the old from before? Or yeah, okay, go ahead.
No, this is a bit different. So I'll just, can I share my screen?
Yeah.
I think, yeah.
How do I do that?
I'm not sure if we need to give you permission, but if you're going to jump to something different.
My thought about what Aidin was saying, yes, I think it would be quite useful to do that. And that is definitely something that we should explore, probably as not just from, say, the perspective of color blindness, but also try to cover other impairments.
I think for the moment to even just make this one very clear point might be good. I have a particular interest in dyslexia, but that's a completely other thing. And like, you know, the Financial Times is the only like broadsheet that's actually done anything about that.
Yeah, no, I agree. I think if we get started with the color blindness, we can then, in the discussion of a paper we write on that, talk about how there are plenty more things that we need to worry about too. But I think if we can build
focused, it might be good.
Right.
Score for accessibility of plots, then we could use that to power a review on the accessibility of these major journals. Like this idea that you just brought up, Aidin.
I don't think it needs, you don't need to do every journal, and you don't need to do a massive time span. But I think you could do something that's systematic, you know, that basically isn't biased. You just took a selection of figures from one or more journals over a specific time period. So that way it's not biasing that, you know, the data were generated in R, the data were generated in something else, or whatever it is. But I think once you have something that's systematic, however it's done, it's basically not biased. And I suppose there's always some bias in things, but you know, I think that gives you data, that gives you preliminary data for whatever you want, where, like for the grant, for the reason of developing this, for the talk, for everything else that you need to do.
That's a good idea. I'm just wondering how to, say, design a questionnaire for that, as in if it's going to be a crowdsourced, as in if we want, as in should it just be an objective metric that we come up with, or should
I would go with, maybe I'm wrong in this, and like there's 18 people on this call, 19 people on the call, and maybe other people have much better ideas. If I were, you know, I would just say get yourself, Dan, and then, you know, a couple of people that are colorblind and even people that aren't colorblind, and just say, how many clusters do you see in this figure? Can you see the cluster groupings? What does this look like to you? And then maybe do some sort of a computer generated picture of what it looks like. But, you know, you don't need to do that for every single one. You just need to have, I think, data to support that this is a problem. And
yeah, I don't know if you would need an IRB for it. I'm just sort of, you know, somewhat joking, but you might need, because it might still technically be human subjects. I don't know.
I think if the subjects are authors on the paper, then they'll be
maybe that's okay.
fine.
I don't know. It's just
you know, if it's just a case of, like, if you want to do something really systematic and comprehensive, then you might need to do a crowdsourcing. But I'd be tempted to just say, right, we just take a 1-year period in 3 journals, search for TISNE or UMAP in the full text, and bring them down and just look at them, you know, and then just see how you're going to evaluate them. And then it may be a case that, you know, you have to actually look at 50 figures, but it's probably doable.
Right.
Yeah.
You just need some sort of criteria that you're evaluating with them or uploading them to some website for evaluation. But even if you upload them to a website for evaluation, I'd probably look at them yourselves, because you're the experts.
I guess probably, I don't know if there are some CAPTCHA services where we could post those and ask, okay, how many clusters in this, how many different colors do you see? As a CAPTCHA, right? As in where people have to just enter how many they see. And
how much impact does, like, the monitor quality have, considering we're now reading all of these journals online compared to printing it out?
Not sure.
I think it's, as in, especially if, let's say, if I have a bigger screen, it would really help, as in, because as I was telling you, that as in the point size kind of matters. So one way is to zoom in, but if, let's say, the figure itself is not a vector image, or if it's not high resolution, then it might be harder to zoom into.
I'm gonna jump in because all these in the talk about online and zooming in. I mean, I think this is all part of the same push, and it could be even more general than just visual impairment. Because the slide that Dan showed about shapes and what he did with Dudosic, that just reminded me of, like, Plotly, for example. So you can mouse over and see, like, data. You can click, let's say, on a legend, and that hides all the points of that match that color, right?
Yeah.
So that's a great way of, like, by, like, elimination process, checking what's happening. You can also zoom in, right? Interactively. So I mean, this is a simple plot. I have more complex plots here from work I did, but, like, I know that, for example, maybe some of these colors on the top right are not distinguishable to you, but maybe like you can click on the legends and try to see what's going on there. Like it would, on the left side here, I'm gonna just run it. This might take a while to load because it has a lot of points, but, like, you can change. Here I have an option on the Shiny app. This is outside of Plotly for completely changing the color scale. And actually the default is a colorblind friendly scale. This other one I don't think is colorblind friendly for a continuous scale where you cannot just, like, click on and off and determine by process of elimination. But basically I think, like, just Plotly gets you a lot of the way there. I think just maybe adding some of this options for having, like, 2 different color scales can help even more.
I think another perk of Plotly is I actually built that into Deshpande as well. I just didn't connect that with colorblindness. But you can convert almost any ggplot object to a Plotly object. They have a ggplotly function. And so
that's what I'm using here. Yeah. Right. And, like, because I mean, here on this website, I actually have, like, static versions, which, you know, have all the problems you were just listing, versus an interactive one where you can, like, mouse over and get more information. So I just think that if you make that the focus of one of your suggestions, right? People love interactive graphics, even if they don't have any visual impairment, right? Because all this information can be really useful. And so,
so like
that's a way of buying in like support from other people that maybe don't see the difference, right?
Right. I think, standardizing that as a means of, say, communication channel parallel or attached to the journal entry, right? Or basically to your paper. So just like we say that the data is available at this place, the code is available at this place. And also,
I mean, I'm going to make an analogy that maybe it's not the best, but I was talking to people at our student conference about sponsorship for the stuff that we're doing in Mexico. And then they were like, okay, you need to what are you going to tell companies? Like, how are they going to help you? And like, one of them is like, okay, we need money. And it's like, this is a charity thing. Another one is like, you tell the companies, like, we're training the people that you're going to hire in the future, right? And so that's kind of what I'm trying to use interactive graphics here for the specific cause of color blindness.
Okay.
Right? So it's like you're trying to be smarter than other people, right? You're trying to like bring them in. I don't know. That's just some thoughts.
Yep. No, that's a great idea, and I think that's definitely something that would, I guess, become more, as an interactive graphics would become more commonplace in the near future, I imagine, as part of the publication itself. I can see that, as opposed to just being a standalone web page where we are just
like Eli, for example, publishes already like HTMLs, or like F1000 was one of the first ones to also publish like interactive graphics. So
I think that's a great suggestion. And I really like the idea of trying to do some sort of like, even if it's just like a 2-page letter, but some sort of review of how good the accessibility of a bunch of recent papers are. And that could be a great suggestion to add.
Yeah, there's a Nature biotech paper, I think, from 2011, 2010. It's just half a page actually by like, I think it's like Casper Daniel Hansen, Jeff Leake, and Rafael Izari, or a subset of them. And it's just like, oh, there's batch effects in this new type of data. And that's it. And that's like highly cited and stuff. Right? So, because it's a starting, you just need a paper as a point to start the conversation. Right?
Yeah.
But people will cite it if it has data. They won't cite it if it's an opinion piece.
Right.
So like Rafa and them, they actually presented different, cases where this was. At the time, there wasn't that many cases. So I think you kind of need to have the data and some sort of stats or something like that. Just it has more strength, I think, if you have something to back it up.
Yeah, that makes sense. And and the idea I kind of had is basically look at, say, these highly cited papers and take their figures and show them how it can be made more accessible, as in just their figures. What could be done to those figures to make it more accessible, and use better colors,
Yeah, I think that would be a great part of it too.
Yeah. To be clearer as to what's happening in the figure.
I'll put the paper I mentioned by Rafa on the chat for the session.
Could you also add a link to your the Plotly page, just as a
sure.
Yeah. We had one other example of a sort of alternative visualization method for something that's often represented as color. So I just figured we might as well go through that.
So yeah, this was basically developed by Melanie Loth in the Fertig lab. So here what we wanted to do is we wanted to see how, say, the gene expression of say 2 genes or whatever, any latent features between 2 cell types or tissue types is changing across. This is from a Visium data set. So spatial transcriptomics data set. So as in here, we just picked two arbitrary genes. So the TN and T1 is shown on the red scale, where the more opaque the red is, the higher the gene expression is, and the PRKCD is on the blue scale, where I guess, yeah, again, the more opaque that is, the higher the intensity or the expression of the gene is. The issue when we were, as in this was, I guess, our first try where we were trying something like this, but then the issue became wherever, let's say, both of these genes are expressed, we go into this purple scale, which becomes really tricky to figure out, as in, is it really more blue or more red, or as in trying
That's specifically a color range that I cannot tell apart.
And it becomes more of a cerebral exercise as opposed to just seeing what it is.
I actually find that quite difficult to tell, and I'm not colorblind.
Right. So what we try to do is basically we just split each of these circles, and each circle or dot here represents a dot on the Visium slide. And each dot was split into two halves of a circle, two semicircles, where the lower half is showing the blue scale and the upper half is showing the red scale. So as in, for me, it's really clear how, let's say, the PRK, as in one of the genes is changing dynamically over the space and the other one is as well. And apparently it's also more interpretable for people with normal vision. So yeah, as in, so this was just an example of the little things that we could do in figures to try and keep the colors apart as possible and not bank on, say, color mixing to provide any meaningful information.
I still find this one quite difficult.
Yeah, I think if the bins were a bit bigger, if we were zoomed in a little bit more, it becomes easier.
Yeah, you need to literally be zoomed in.
Mm-hmm.
Do you think, actually, that's an interesting point, do you think that spatial data visualization is a more difficult task than TISNE and UMath?
So I think there are problems which are common, as in which are not just about color blindness in TISNE and UMath, where we are talking about overlaps and all of these things, right? Spatial, probably you don't have those problems. Everything is separated by itself. So I think the disadvantages are largely color blindness based in spatial, as in, and if, let's say, you have, again, 20 different cell types based on what kind of immune cells or cancer cells they are, then you might run into the same problems as you might in TISNE plots.
So for that last example where you're literally just looking at two cells,
yeah,
like why not black, white, and gray?
Yeah, you could do that as well, yeah. And probably color is prettier, I guess.
I was wondering, actually, so like how many of us have actually had like a data visualization class? I mean, I feel like I've learned things. I mean, I was a biostatistics PhD, so I learned some things in my classes, you know, kind of general things. And then, I mean, I learned a lot from my advisor and my advisors and, you know, sort of, you know, best practices from different people. But I don't know. I mean, it's one of those things like if, you know, there's like certain things that, you know, if you can introduce them in a curriculum, then, you know, that's a way to just sort of make sure that those things are like, you know, ingrained, right, in like students' minds. And so I realized like when I'm mentoring students, like I try to remember the colorblind issue, but because like I don't have it, I think at the very end, you know, it's like first you just use like the default colors in ggplot. And then at the very end, it's like, oh yeah, we should actually, you know, also try to make it colorblind friendly. And it's just, I mean, and I've been trying to get better about that, you know, when I prepare plots for publications, but it's not something that's, you know, really the first thing that I think about. And it's also not like something that I was sort of, you know, like taught formally. So I think it's one of those things where, you know, kind of like developing, I don't know, like educational material somehow. It's like, how do you kind of, you know, like put it, you know, so that it's in people's, you know, the forefront of people's minds. And it's sort of like.
That's a good point. I have never had any formal education in, I guess, graphics or.
I have not either.
Yeah,
I think even if you had, things are changing really fast. Like one of the things, I've been to like some of these bigger data, there's a whole data visualization things at different stages and stuff. And the things that they might have talked about a few years ago are now different. And even if you look at, like, we'll say people that are doing data visualization for things like, you know, BBC, Financial Times, you know, New York Times and things like that. Though, Rafa's actually got a couple of those people into the department to give talks. Mm-hmm. Things are changing, and they've changed a lot. And the figures that, you know, people presented a couple of years ago are not the visualizations that are being presented now. And I think if you look at any big visualization that's just in like New York Times, they normally have now like a web front end to go along with it. So, and a lot of that is done through R. So I think things are changing a lot. Mm-hmm. And really what's important is the defaults are a good default. And that people need to be aware that their packages should have reasonable defaults. So you need something that basically raises the awareness and then provide those basic tools such that like the default is always relatively reasonable.
Yeah, I was wondering if as part of, say, bioconductors conferences itself, and there should be, I guess, a concerted effort to try to have a session about, as in where someone would explain from a usability perspective or user experience perspective as to how to design your defaults or how to design your, because both even Dan and I are kind of consumers in a sense, right? And we are trying to learn it on the way, but I would assume there are people who are, especially in, I think, the field of GIS and as in where a lot of the information goes out to general public, they have thought a lot about accessibility and interpretability of their figures.
Well, the way the conference is organized, it's you volunteer. So volunteer, propose the track, identify the speakers, and you know what? It'll probably happen. So like last year, it was just a call. Anybody who wants to help. Okay. You know, it's whoever signs up to help gets to. This isn't a top down. This is literally like join.
No, yeah, that's.
If this is, you know, if this is important, then, you know, the best person to drive it is you.
Okay. No, that's.
So join the BioC 2021 conference committee, find the speakers that you think that would be good speakers, and and just put it out there and just go for it. Yeah, definitely.
Yeah.
Join the community advisory board too.
Yeah.
Trying to point at it, but it's a bit hard.
Yeah, I mean, I think I've been in communication with the CAB, and I've been thinking about that already a little bit. So I might just do that if I can. But I think we're towards the end of the time, so I just wanted to, I think I'm sharing the wrong thing. I wanted to just come back to this Google Doc again and just encourage, where did my Zoom go so I can share? Encourage people, if there was a particular you would be out with, and it can even be helping out with the paper itself to just sign up here.
Can you post that in the Slack channel or various different places?
Yep. Yeah, the link. Yeah, I can post it in the Slack too. The link is currently in the chat for this session.
And once this is into kind of like a project rather than just an idea to talk about, I would definitely make a Slack channel for this as well. Mm-hmm. So people can join and contribute or do what you need. So make a Slack channel for this. Anybody can make a Slack channel. And then, you know, organize around that. That would be a good way to go forward in this. And
I think I'll make a channel called Accessible Viz, because there's already Cool Viz, and that would be the counterpart.
Maybe we can say the coolest Viz.
Everyone will be comfortable sharing their emails on a doc that might be good. You know?
Yeah, maybe we don't need that column. Just if you want to, you can just give your name, and then especially if you're already in the Slack, that's totally fine.
I posted a summary of my ideas on the chat of the session on the Path Level website, but I'll post it on Slack once you make that channel.
Awesome. Cool.
Thank you for driving this.
Yeah.
Thanks for
next
getting
year.
it started, Atul.
You have what's it called?
Yeah, I'm glad this kind of came out of that beers meeting discussion we had, so that's actually really cool.
Yeah. Hopefully it just keeps snowballing into bigger and bigger things. And
Thanks everyone for joining, and I'm excited for what we're going to do next.
mm-hmm.
Thank you for organizing.